Hello, I'm Jan Leifman.
Hi, I'm Gayatri from Menon.
Welcome. Welcome to Med News Week conference, where we feature presentations by medicine's global leaders. Today we have an amazing keynote speaker in Dr. John Shin. Dr. Shin is an assistant professor in medical oncology, hematology division at the Loma Linda University.
In today's presentation, Dr. Shin discusses the most recent advancements in microbiome and cancer immunotherapy and where its research is headed.
Did you know that Dr. Shin is a globally renowned researcher who is leading clinical trials to study the role of nutritional interventions to improve cancer therapy efficacy and its effects on the gut microbiome? He has also written a practical guide assessing the effect of using exercise as a prescription for cardio-oncology rehabilitation.
Such a rising pioneer in the field of medicine.
So let's tune in. Let's tune in and learn from this rising star.
So let me begin by asking the question that might be on top of some people's minds of what is the microbiome? And so very simply, the microbiome is all of the bacteria that live in your intestines. So the totality of these bugs in your gut is known as the gut microbiome. And the interesting fact that a lot of people may not be aware of is that there's approximately 1 trillion bacteria that live in your body. And this outnumbers the number of human cells in your body. So it will be accurate to say that we are more bacteria than we are human. And so for the longest time, we felt that this biomass, this bacterial count, was an inert thing that just kind of co-inhabited with us in our bodies. But we are understanding now that rather than being just passive standby organisms, the microbiome actually intricately affects your health in many aspects of life. So in the last decade or so, it's become really clear that the gut microbiome actually actively modulates many different dimensions of health. And in this review, they went into some of the nuances, but you can see that the functions of the gut microbiome, it influences, among other things, developing the immune system, neurologic signaling, synthesizing vitamins and hormones, metabolizing the various dietary components. And as a result, if you have a disordered microbiome, this has been associated with a host of diseases, including neurologic, psychiatric, respiratory, cardiovascular, GI, so on and so forth, and obviously in cancer as well. Here we see that the gut microbial diversity, so the different types, how many different types of species you have living in your intestines, it starts to increase from the moment we're born. It peaks right around when we become teenagers, and then starts decreasing as we advance with age. And a lot of things impact this diversity, which we'll get into in a bit. Now, here's a study of diet and how it shapes our gut microbiomes. And what this team did was that they studied the diets of children living in rural Africa, in the country of Burkina Faso, and compared that to the diets of children in Europe. And then they compared the microbiomes to see how different were the microbiomes of the two populations from each other. And what's interesting about the diet of these rural Africans was that they tend to be low in fat and animal protein, rich in starch, fiber, and plant polysaccharides. And it's predominantly vegetarian, which makes sense. And what they found is that there is a very big difference in what the microbiomes look like of these two populations. So in the African population, you have a predominance of what we call Bacteroides. And the Firmicutes, that type bacteria, make up a relatively smaller proportion of this. Whereas in the European children, you see that it's completely flipped. You see a predominance of the Firmicutes and a relatively low contribution from the Bacteroides species. Now, why is this important? Well, the bacteria that live in your intestines determine what kind of metabolic effect that they have on your system. And they found that in these children who live in rural Africa, the total number of short chain fatty acid that they produce as a result of their microbiomes was far greater than their European counterparts. And the short chain fatty acids that are made in the gut microbiome are one indicator of gut microbial health. And we found that they actually play a big role in modulating some of the health responses of the microbiome. There's a lot of things that go into the microbiome, and there's a lot of metabolites within the microbiome that impact your overall health. But short chain fatty acids come up over and over again as being one of the things that are most important in terms of metabolic byproducts of your microbiome. So the important thing to note is that between these two groups of children, the ones who adhere to a higher fiber plant-based diet, a non-Western type diet, not only had a big change in the type of bugs that lived in the microbiomes of these people, but that they actually detected a metabolic difference in these microbiomes. Now, the way that the microbiome modulates health is very, very complex. It's exquisitely complex, and we're only beginning to scratch the surface of understanding how it works. But we know that the production of certain metabolites, such as what I just mentioned, short chain fatty acids, is associated with a more healthy microbiome. And in general, the more diversity that there is among the gut bacteria, the better. And so you'll hear me talking about gut microbial diversity over and over again. And you can see various different factors play a role in causing what we call a disordered gut microbiome or gut dysbiosis, or a healthy gut microbiome or gut eubiosis. And then depending on whether your gut microbiome is healthy or unhealthy, it can upregulate or downregulate certain things and modulate various aspects of health. So as I mentioned, the diet is probably the single largest mover of the gut microbiome. And in general, we're seeing that the Western type diet that's high in animal protein, that's high in saturated fats, that's low in fiber, tends towards gut microbial dysbiosis. Whereas a higher fiber diet that's rich in plant foods, that's lower in animal proteins and saturated fats and things like that, tend towards creating a eubiotic or healthy gut microbial state. Now, diet isn't the only thing that moves the needle when it comes to our gut microbial health. It turns out that the amount of exercise that we do also impacts the gut microbiome. So here's a study that looked at the gut microbiomes of professional rugby players. And what they found is that among these professional athletes, when they actually compared them to BMI match controls who are sedentary, they had more diverse microbiomes, and the microbiomes produce more short-chain fatty acids, which is what this represents here. So the athletes compared to the low BMI controls. And so in these studies, they also controlled for food as well. Here's another study that shows that exercise really alters the gut microbial composition. And what they did was they found lean and obese participants and matched them up. And for 6 weeks, they asked both groups to exercise. And so at baseline, prior to exercise, this is just a principal components analysis, but basically you can see that after 6 weeks, their gut microbial diversity changed, the composition of their microbiomes changed. But what's interesting is that in this study, after the 6 weeks of exercise, all participants were actually told to go through a 6-week washout period when they were completely sedentary. And the sedentary 6 weeks seemed to undo essentially everything that the prior 6 weeks of exercise did, which lends more credence to the fact that it was indeed the exercise that moved the needle in the microbiomes. And so you can see that the composition of the gut bacteria in both the lean and obese populations changed after exercise, and then they reverted back to what they looked like prior. Here, when you look at the short chain fatty acid production of the gut bacteria, you can see that the lean people here actually benefited more. Their gut microbiomes increased short-chain fatty acid production more than their obese counterparts, but even the obese people increased their production. But in this smaller graph in the upper right-hand corners, you can see after the 6 weeks of sedentary washout and lack of exercise, you can see all of their microbiomes, the short-chain fatty acid production is reverting back to their baseline levels. So this was pretty strong evidence that indeed physical activity changes your gut microbial composition. Now, I want to add the caveat here that there are many, many, many other factors outside of diet and physical activity that change your gut microbial composition. Things such as your stress, the amount of sleep that you get, geographically where you live, all of these things affect your gut microbial composition. But I'm going to just focus on these two for now because it gives us a smaller scope to work with, and it also gives us two of probably the most modifiable lifestyle factors that we have direct control over when it comes to our own personal lives and the lives of our patients. So I want to switch gears now and talk about cancer immunotherapy. And before I can do that, we have to define what is cancer immunotherapy. And in order to do that, we have to understand the cancer life cycle, if you will. So here is the cancer life cycle, and the immune life cycle begins something like this. So if you have cancer cells that end up dying for one reason or another, whether they were actively destroyed or maybe they died as a result of outstripping their blood supply, when they die, these cancer cells release antigens that are presumably, hopefully, foreign to the body. These antigens are picked up by what we call professional antigen presenting cells. Dendritic cells are the most important component of this population of cells. And these antigen presenting cells scoop up the antigens and traffic to the nearest lymph nodes, where they then present these antigens to T cells and help activate the T cells. This is called a priming stage. Once the T cells identify these antigens as being foreign and they become activated, they're released into the bloodstream and they traffic to the tumors using chemokines. They infiltrate the tumors and then they go forward to hunt for the tumor, if you will. And once they recognize the tumor as being the target that they're looking for, they go ahead and kill the tumor cells, which then release more antigens, and the entire cycle starts over again. Now, in this process, in this cancer immunity cycle, there are factors that can sustain the cycle by providing more stimulation, or there are factors that can inhibit the cycle and allow immune escape or immune evasion on behalf of the tumor cells. And you can imagine that at any of these steps, something can go wrong and the tumor can learn to overcome immune detection and therefore continue to grow unchecked by the immune system. And the goal of cancer immunotherapy is to address whatever kink in the road is blocked by the tumor, or to overcome some of these hurdles, or to make them more effective, so that the end result is that the cancer cells can be detected more effectively by the immune system and taken out by the immune system. So all of the different technologies that we've developed to address the cancer immunity cycle in any of these steps as a whole, as an aggregate, is known as cancer immunotherapy. And there are many, many different forms of cancer immunotherapy. But for the purposes of tonight, I'm going to just talk about the most well-known category of cancer immunotherapy, which is the immune checkpoint inhibitors. So here are the names of the most common immune checkpoint inhibitors that we know of today. And these immune checkpoint inhibitors utilize an immune checkpoint known as PD-1 and PD-L1. So PD-1 is a receptor that's found on your T cells, and PD-L1 is a receptor that's found on the surface of many cells in your body, including tumor cells. And you can think of them as brakes on the immune system. So this is a way of natural cells in your body that are good cells. This is a way for them to signal to your immune system, don't kill me, I'm one of the good guys. However, we found that many different tumor cells are able to take advantage of this system so that they can evade immune detection. And so when you have a cancer antigen presented in the context of the major histocompatibility complex and the PD-1, PD-L1 system is engaged, it sends an inhibitory signal to the T cell, and therefore the T cell doesn't take out that tumor cell. Well, these checkpoint inhibitors are antibodies that block this from happening. So you have PD-1 inhibitors such as nivolumab and pembrolizumab that block the receptor here at the PD-1 spot, or PD-L1 inhibitors that block PD-L1. And when this doesn't happen, it's essentially like taking the brakes off the immune system. And so the T cell is able to then go ahead and kill the cancer cell because there's no inhibitory signal being sent. Now, there are many other different types of checkpoints, immune checkpoints out there. PD-1, PD-L1 is only one of them. You have CTLA-4, which was also made famous, but we're finding that there are literally dozens out there. And I would be remiss if I didn't talk about just how great of an impact these immune checkpoint inhibitors have had on cancer immunotherapy, because up until this point, in the decade or so preceding the development of immune checkpoint inhibitors, immunotherapy research was languishing. And the number of scientists committed to researching the area of cancer immunotherapy was dwindling. But once the immune checkpoint inhibitors came on the scene, it really revolutionized the field. And now this is one of the fastest growing areas of research. Public interest has exploded because of high profile cases such as our former president, Jimmy Carter, saying that his metastatic brain melanoma was cured thanks to immunotherapy. And because of the great changes and the great leaps forward in treatment that immunotherapy has offered the world of cancer care, in 2018, the Nobel Prize in Physiology or Medicine was jointly awarded to Dr. James Allison for his work in discovering CTLA-4 and to Tsuku Honjo of discovering PD-1. And both of these are the most common and most well-known immune checkpoints, as I mentioned before. So then, let's talk about now the gut microbiome's impact on immunotherapy. Well, we had a suspicion for a while that this may be the case, that your gut microbiome can impact response to immunotherapy. But this really came to the fore with Dr. Penato and his group's observation that if patients who went on to receive immunotherapy received antibiotic treatment within the 30 days leading up to the start of their therapy, they had worse outcomes with worse survival. And this phenomenon was initially thought to be due to the fact that, well, the patients who get antibiotic treatment before getting cancer treatment obviously must be suffering from some kind of infection. Obviously, they must be sicker. Obviously, they must have a lot more other comorbidities, and therefore that's probably why their outcomes are not as good. But what this study has shown is that even when you account for these things, even when you control for these things, still you can see that there is an overall survival detriment in the patients who received antibiotic therapy leading up to their exposure to immunotherapy. And this really caught the eye of a lot of people, because now we realize, hey, a disordered gut microbiome is not simply a marker of bad outcomes, but actually may actively be driving the bad outcomes. And as you know, antibiotics it's like a nuclear bomb going off in your microbiome, and it'll be like the gut microbial holocaust. And so nothing will really disorder the microbiome more than antibiotics. So when we saw this phenomenon happening, we said, well, there's more than meets the eye here. And since then, there have been dozens of studies literally that study this phenomenon, that the gut microbiome actually impacts anti-cancer therapy, both with immunotherapy and traditional cytotoxic chemotherapy. So here are just a smattering of studies that have shown that immunotherapy treatment efficacy is impacted by your gut microbiome. And then there are a bunch of studies here that show that immunogenic chemotherapy is also impacted in its efficacy by the gut microbiome. Now, this is a study that was published out of Jen Wargo's group at MD Anderson, and they really have kind of pioneered this field of studying the link between the gut microbiome and its ability to modulate response to checkpoint inhibition therapy in melanoma patients. And in this study, they found that patients who get immunotherapy for their melanoma and end up responding well, these patients tended to have a higher gut microbial diversity than the patients who ended up not responding. So you can see here you have high, intermediate, low gut microbial diversity. And in the blue, the patients who had higher diversity, you can see that their progression-free survival was superior to those with intermediate and low diversity. And you can see the score of their gut microbial diversities here. Now, looking at this chart, some people might say, well, John, how do you know that gut microbial diversity is not a marker of response as opposed to a causative agent? So maybe it's correlated with response, but it's not causative of the response. And to test that principle, they actually took germ-free mice, which are mice that don't have any microbiomes, and they transplanted some of these mice with stool from responders, and then a different group of mice, they took stools from non-responders. And then they grew melanoma cells on these mice. And then they exposed them to immunotherapy afterwards. And what they found is that the mice who were transplanted with stool from the responders, their tumors responded much better to immunotherapy than the mice who were transplanted with stool from the human non-responders to immunotherapy. You can see that the tumor sizes and the increase in change. And you can see the tumor volume over time. In the mice bearing the stool of the non-responders, their tumors are taking off, whereas the tumor growth is much more attenuated in the mice who had stool from the responders. So this was an exciting kind of proof of principle that, well, the gut microbiome actually does modulate response to immunotherapy. And you might say, well, John, that's all and good in mice. What about in humans? Can this be reproduced in humans? And the answer turned out yes. So last year, we had 2 landmark studies published in the journal Science that showed conclusively that when humans are transplanted with stool from other humans who actually responded to immunotherapy, these people can increase their odds of responding to immunotherapy. And in both of these studies, they took patients who had melanoma, who stopped responding to immunotherapy or were non-responders from the beginning. They never responded to immunotherapy. And then they transplanted these patients with stool from patients who were known to respond to immunotherapy. And they were able to convert quite a few patients in each group and turn them into responders to immunotherapy. And it was just a stool transplant alone that was able to accomplish this, which is phenomenal. Just recently, Jen Wargo's group also published this paper that showed that the dietary fiber and probiotics also greatly impact the gut microbiome and ultimately its response on immunotherapy. So here they found that those who had the highest dietary fiber intake their survival probability was higher than those who had insufficient fiber intake. And when you look at the profiles, here's a heat map of RNA sequencing of CD45 tumor-infiltrating lymphocytes. You can see that the T cell activation, there's a higher expression of genes related to T cell activation and interferon response in mice who received a high versus a low fiber diet in the setting of being treated with an anti-PD-1 checkpoint inhibitor therapy. Now, what was really interesting here is what they found with probiotic supplement use. So this shows the survival probability of people who had probiotic use versus who did not. And you can see in the dark purple, the people who did not have probiotic use, it seems like their survival curves were higher than the ones who actually had probiotic supplement use. And in here, you can see that when you have those who had dietary fiber, sufficient fiber, and no probiotic use, these had the highest survival advantage. But the moment you took even the population of patients who had sufficient dietary fiber in their diet, when you threw probiotics on it, now their survival benefit seems to have been completely erased by virtue of the probiotics coming on board. And so what this tells me is that it's a cautionary tale, because when we think of the microbiome, a lot of us gravitate towards probiotics and we say, well, let's go ahead and give our microbiomes a jumpstart by taking probiotics. But the way that you obtain over-the-counter probiotics these days is a very blunt tool. The manufacturer of probiotics, well, first of all, probiotics aren't FDA regulated in any way. The manufacturers of probiotics simply put in whatever they think is fit for their product. And there's no agreed upon standard of what species of bacteria, how many colony forming units, and in what proportions. And so if you blindly just simply supplement with probiotics, it can actually be detrimental if you don't know what you're doing. There are many studies going on right now trying to tease apart exactly what bacterial species are the most beneficial, in what proportions, or, is this something that might be a little bit more tailored to the individual, where a certain bacterial cocktail might help one person but actually harm another person. And so this is in the field of active research, and I believe that in the years to come, we will actually be able to develop intelligent probiotics that will be beneficial to you prior to undergoing immunotherapy. But as it stands, the probiotics that you take off the shelf are actually not designed with the individual microbiome in mind. And I would say buyer beware, because they can actually do more harm than good. So this leads me to the research that I have going on currently. Because of all this data that I shared with you about the effect that diet and physical activity can have on the microbiome, and because we know that the gut microbiome, when it's optimized, can increase the odds that you respond to immunotherapy, we wanted to tie this all together in the context of a single trial. Now, there's a couple of things novel about this trial. One is that we are looking at the impact of diet and exercise combined on the microbiome. And as of yet, I do not believe that there's any other trial that has studied both of these factors at the same time. The other is that of the trials that have studied the impact of diet on the microbiome, there have been several controlled feeding studies where the investigators provide all the food to the participants. And this is important because it provides the purest intervention, and it provides a great proof of concept of how we can change the microbiome in response to immunotherapy. However, in terms of generalizability, it's not very good, because because in the community, doctors cannot cook for their patients and provide 3 meals a day. And so you can't really take that knowledge and say, okay, well, I'm going to provide the same meal for my patients. So what this study looks at is the efficacy of a diet and exercise prescription, where we tell patients, this is what you should eat. These are the guidelines you should follow, and to see how well they're able to implement that in their normal day-to-day life. So the title of our study is called The Effect of Diet and Exercise on Immunotherapy and the Microbiome, or it's EDEN for short. And what we do is we randomize patients who have melanoma that cannot be resected or are metastatic at diagnosis, and we randomize them 1-to-1 to an intervention arm or a control arm. And in the intervention arm, patients will be given a diet prescription where they're told to adhere to a high-fiber plant-based diet. And we have a team of dietitians on board to help them change their diets and to change their cooking patterns so that they can meet these dietary targets. And also, we give them an exercise prescription, which is simply the CDC guidelines for exercise for people across the board, which comprises of 150 minutes of moderate intensity exercise or 75 minutes of vigorous intensity exercise per week. And we monitor their exercise activity by sending them a fitness watch, a fitness activity tracker. We have people log whatever they're eating several times throughout the trial, maybe about 3 days in a row on 4 separate occasions. And we do monthly unannounced food recalls where they recall for the last 24 hours what they ate. And they enter it into an app on their smartphones called MyFitnessPal. And this is how they get their nutrition information to us. Now, patients who enroll to the control arm, they will simply be given general direction on healthy eating and healthy living, but they will not be asked to adhere to any specific diet or exercise prescription. And patients in both groups will be undergoing treatment by an immunotherapy combination of relatlimab and nivolumab. Now, this is hot off the press because relatlimab is a LAG-3 inhibitor. LAG-3 is another immune checkpoint that is very novel. And as of this past Friday, March 18, the FDA has approved the combination of relatlimab and nivolumab for use in treating first-line melanoma patients who are unresectable or metastatic. And in our trial, we'll be obtaining baseline stool samples and stool samples at various time points along the way to detect if their microbiomes have changed. And then we'll be assessing whether or not the microbiome's changed. And if there is a movement in the microbiome towards greater gut microbial diversity or other signs of eubiosis, it'll be helpful to see if there's also at least a numerical trend towards a higher response rate in the patients who've changed their microbiomes. Now, the primary endpoint for our trial is feasibility. The reason being that this is a decentralized clinical trial. So this is a novel form of clinical trial where everything we do regarding a trial is completely virtual. So all of our patients we will meet online only, not in person. And everything is all trial-related activities will be conducted via telemedicine. So this is great for the patients because they can enroll from anywhere in the country. They don't have to travel to participate in our trial. And in order to meet with our dietitians or talk to any of the people on our study group, they simply log on and have a conversation. All of the data we collect is done electronically. And as a result of being on this trial, participants will not have to undergo any extra testing or scans or anything like that other than sending us the stool samples periodically, which means that they can continue their treatment with their local oncologist. This will not get in the way of any of the treatment plans that the oncologist has for them, and they can still participate in our trial in the background. And so we're looking forward to seeing the data that this study generates because it's not only a study about diet and exercise and its impact on the microbiome, but it's also a study of the proof of concept of a decentralized clinical trial whereby we see, will this study design enable us to answer the question that we need to answer? Will it generate evaluable data? Ultimately, are patients adhering to what we asked them to do and sending us back good usable data? And so our primary endpoint is feasibility to see if there's compliance with the requirements of the trial. And as a secondary endpoint, we'll be measuring overall response, progression-free survival, and quality of life. And then we'll have a bunch of exploratory microbiome analyses done, which includes a whole genome shotgun sequencing. So if you are a patient with melanoma, or if you are a provider who sees melanoma patients, and you're planning to start someone on relatlimab or nivolumab, or if you're a patient who's about to start on that combination, feel free to get in contact with us to see if you qualify for our study. You can send us an email at nciedenstudy@mail.nih.gov, or you can call us directly at 240-858-3525. And so we're excited to see enrollments from across the country.